Hi everyone, my name is Simran Kaur and I'm working with Professor John Christodoglou at Murdoch Children Research Institute, Melbourne, Australia. I would like to start by saying a big hello to all the lovely KIF1A superheroes, their families, and all of my KIF1A colleagues in the Research Roundtable Network. Thank you so much KIF1A.org for giving us the opportunity to be a part of this amazing well-knitted KIF1A family, and we are very grateful for that. So today I'm going to tell you about what we are doing on our part to uncover the hidden drug to benefit our loving kids and adults battling each day with KIF1A associated neurological disorders, or simply KAND. KAND is a severe brain-related disorder that, along with affecting brain and behavior, also affects multiple other organs such as eyes, muscles, bones, stomach, and urinary system, thus significantly compromising the overall quality of life. Although each affected individual manifests different combination of symptoms, but the most common features observed are development delay, intellectual disability, abnormal muscle tone, spasticity, and seizures. During past few years, the phenotypic spectrum of KAND individuals has been further expanded to include other neurological conditions, such as PIHO syndrome. And our research has further uncovered the overlap with Rett syndrome, which is a rare X-linked neurodevelopmental disorder. Now let's quickly refresh what KIF1A does within us. The human brain is a very complex organ made up of billions of cells called neurons that communicate with the neighboring cells through very specialized junction points called synapses. It helps to synchronize the brain and body function. So in terms of the structure, the neuron consists of the dendrites, which is like these little projections here, the cell body, the long axon, and finally the neurite terminals. Now we can consider the cell body of the neurons as the factory where most of the proteins are synthesized within the cell. Now these newly synthesized proteins are then packaged as little cargoes, which are then transported along the axon towards the neurite terminals in a very rapid and well-coordinated manner. Now, any small holdup in the delivery of these critical cargoes can disturb the logistic chain of events which can ultimately affect the normal function of the brain. Now, KIF1A is one of the motor protein that helps in transporting these key cargoes from the cell body towards the synapse. It's a dimer. That is, it consists of two chains that are twisted like a cable in electrical wire. There are two heads, which are also known as the motor domains, that walk step by step on the microtubule protein network using the ATP molecule as the fuel source. Now this beautiful animation might be familiar to you, but it's a great way to see KIF1A in action within the cell while transporting a very bulky cargo. The two motor domain walking step by step on a very long thread-like structures called microtubules over a very long distance. So we can sum up as efficient KIF1A movement results in healthy synaptic function that further ultimately results in proper brain function. In our cells, specific proteins are encoded by specific genes. In our case, a normal KIF1A gene results in a normal KIF1A protein that undergoes a normal movement within the neurons and results in healthy brain function. However, in case of KAND individuals, due to small spelling mistakes known as mutations or variants within the gene, a defective or variant KIF1A protein starts building up within the neurons. Previous research have shown that although the variants can occur within the whole of the KIF1A protein, however, the head or the motor domain is the hotspot region for variants to occur. So far, research has shown that as opposed to the normal movement of KIF1A, the severe variants within KIF1A motor domain either results in non-binding of KIF1A with the microtubule network or results in slow speed or no speed at all for KIF1A, causing traffic jam-like situation within the neurons. At the moment, there are no specific therapies for treatment of children with KIF1A abnormalities, with only symptomatic management currently available. Overall, the therapeutic strategy for our KIF1A research mission is to develop therapies to benefit this generation of KAND individuals. So the roadmap highlights targeting multiple ways, such as exploring the small molecule-based research or gene-based approaches at one time to achieve this goal in a very timely manner. Our research focus is centered on small molecules. Most of our everyday drugs, such as aspirin, antihistamines, etc., they're all made up of small molecules. Apart from the naturally existing ones, there are essentially two ways to prepare new ones. One way is to start from the scratch and stepwise combine the building blocks in a chemistry lab to build up the final molecules. It's much like what our kids do while they play with the Legos to make their favorite shapes. But this process is very expensive. It takes more than 15 years to come to the market, and provided there is a lucky one in the draw exhibiting the desired safety and efficacy profile, because the success rate is very low when it comes to the FDA approval. A second strategy, which is currently widely used, is called drug repurposing strategy. In a simpler term, it means drug recycling. That is to test whether any of the existing FDA-approved drugs, often prescribed for any other health-related conditions, can correct or at least improve the molecular function of KIF1A. Depending upon whether we find any interesting compounds, this route is comparatively faster, affordable, with higher FDA success rates. Even in the drug repurposing, either we can manually screen individual small molecules that have been reported so far in literature one at a time, or we make use of robotics to accelerate the process, which is usually known as the high throughput screening. This technique has been demonstrated to be very powerful and exceptional method to identify small molecule for therapeutic applications in a very time efficient way. With this technique, we can screen very large drug libraries using the special plates and instruments in a much shorter time frame. Since high throughput screening is a robotic based system, so we need to establish an assay in a lab that is compatible with the fast screening method. Since KIF1A is mostly found in neuronal cells, it creates a major practical challenge to study it directly in the cells derived from the individual's brain. To address this challenge, we have developed a simple and fast COS7 cell-based model that will be used for our screening purposes. Briefly, COS7 cells, they are monkey kidney cells, and since KIF1A is absent in COS7 cells, so we need to introduce a synthetic DNA sequence that will encode for a very small part of KIF1A protein. Now, this this particular truncated KIF1A protein will only consist of the motor domain and a little part of the rest of the KIF1A molecule, because our interest to study here are the variants that affect the head part or the motor domain of the protein. Now, this truncated KIF1A is tagged with a tag which is known as M-citrin. Now, this M-citrin, it emits out a bright green light under the microscope, and thus it helps us to localize the position of the expressed KIF1A protein within the cell. For example, the normal KIF1A has a tendency to accumulate at the boundaries of the cell, as shown in the schematic here. However, most of the variant KIF1A, it remains stuck within the cell body. It signifies that the variant KIF1A is showing a defective movement within the cell, which is why it is unable to move up to the boundary of the cell. So this is what it looks like under the microscope, where the blue color shows the nucleus, and the green shows the localization of expressed KIF1A protein within the COS7 cells. So using this model, we can test the effect of small molecules on KIF1A's localization within a dish in a lab environment. So we will be taking videos of the living cells expressing variant KIF1A for about a day, which are then later treated with small molecules for another 3 days to find out which of the small molecules from our chosen library can help the KIF1A molecule to move within the cell. After developing and validating the assay, we will be using a well-characterized FDA-approved small molecule library from an organization called Compound Australia. This library consists of about 3,000 small molecules, which are often used in the other related health conditions. The idea behind to choose a small library is to first show that the assay that we have developed actually works on the robotic system before we go ahead and dig deeper with the other larger screens. For the analysis, we will examine the effect of small molecules on the localization of variant KIF1A by analyzing the green signal which is coming out of the KIF1A protein. So the idea is to find small molecules that can reverse the KIF1A defect and make it move within the cell. The next two steps involves a careful selection and validation of small molecules with desirable effect on KIF1A molecule for further study. In the modern time, virtual screening of small molecule libraries is an alternative and complementary approach to lab-based high-throughput screening of molecules to accelerate the identification of small molecules during the drug discovery process. So in this study, the screening of drug-like compounds from a publicly available databases such as Chembl is used to examine the effect of molecules on the chemical structure of KIF1A using computer-based programs. Using this approach, AMRI's research team in collaboration with KIF1A.org has identified over 100 small molecules that have been virtually observed to improve the function of KIF1A. We aim to synthesize these chemicals and test them in the lab using our developed KIF1A cell-based assay. We think this is an excellent collaboration that will expedite the selection of relevant small molecules for further research. Now, while we are trying to find small molecules from the FDA-approved drug libraries and other drug-like compounds, but what about the existing kinesin modulators? Although there are no KIF1A specific modulators that exist in literature so far, but we can use the fact that the structure and function of the motor domain of kinesin superfamily members is very similar. Now, when we check the literature, these 5 small molecules have been previously shown to change the function of other KIF motor domains. And we are now investigating if they will change KIF1A motor function too. Ultimately, we would like to see if our chosen small molecules does show an effect on the neuronal cells which are derived from KAND individuals. For this, we will use induced pluripotent stem cells, or simply iPSCs, that are derived from the skin cells of the individuals. Now, these cells, they have the capacity to become any type of the cell within the body. For our interest, we will force the KAND individual iPSCs to make mutation-carrying neurons. At the same time, we need a corrected neuron expressing normal KIF1A. We call it as an isogenic control. These cells will have the same genetic background as KAND individual mutation-carrying neurons, except that they will express normal KIF1A and will serve as a control for the analysis purpose. Since we want to see the effect of small molecules on the movement of KIF1A, so we need to again add a green label to help us visualize in the lab. And this time, instead of tagging the KIF1A, which is coming from the KAND individual cells, we will tag the KIF1A specific cargo instead. Ultimately, KIF1A is going to carry the green labeled cargo, and if our selected small molecule is working, then the variant KIF1A will show the speed and localization of green cargo similar to the normal KIF1A. So overall, our aim is to develop high throughput screening based approach for KIF1A therapeutics and test the FDA approved libraries, small molecules identified using computer programs, and other KIF1A modulators to identify small molecules that can then be further optimized and taken into further clinical studies. While finding therapeutics is absolutely critical, we also want to keep working to improve the timely diagnosis of KAND in our kids. This can be done if KIF1A gene is included in every possible panel that are routinely used for the diagnostic testing. In Australia, PanelApp is routinely used during genetic diagnosis, and we have been successful in adding KIF1A to a list of targeted genetic panels. Moreover, we need more family-oriented resources that could help them guide through the initial journey after a confirmed diagnosis. It is important to connect them with the relevant clinicians and KIFIA.org for further support. So we have worked together with New South Wales Health Centre of Genetic Education, Professor Wendy Chung and Leah Boyle, to develop a fact sheet for our Australian families to help them guide further after the KIF1A diagnosis. Last but not the least, we do need to make more noise to attract media attention so that our research network can go further, and so does the opportunities for further financial fundings to help support this critical research. In terms of the significance of this project, I would like to say that our high throughput drug repurposing strategy will prove to be cost and time efficient way of identifying targeted treatments for children with abnormalities in KIF1A function. The specific outcomes of this research will have a critical impact on affected children and their families. It is also noteworthy that pathogenic KIF1A variants cause a range of other related neurological disorders such as spastic paraplegia, Thus, it is very likely that therapeutic candidates we identify through our high throughput screening platform could be more widely clinically available. In the end, I would like to say a big thank you for our wonderful international collaborative team and Research Roundtable Network members for all of their support and guidance in developing this project. It wouldn't have been possible without the generous financial support from Murdoch Children Research Institute and KIFIA.org. We are hugely indebted for all the trust and love of all our wonderful KIFIA families. Thank you so much. And if you have any questions, please do join us during the question and answer session. We'll look forward to see you there. Bye for now.